Hey Science Peeps, this is Mr. F for Birdman Science Lessons, and this is the first installment of a four-part series on the scientific method, this of which is going to be the scientific method, parts, and use, followed by experimental design and identifying variables, and we'll do some practice in that segment as well. And from there we go on to experimental questioning and hypothesis formatting, and the three different types of hypotheses, and how to write a very good scientific question and hypothesis depending on what type of experiment you're conducting. And then lastly, we are going to cover scientific article writing or lab report writing. So this is going to be all the product of all the great work you do in your experiments. Okay, so the operative question that we're going to have here is, how do we gather scientific information? What are the different modes, or are there even modes or processes that we follow to do this? And that's what this segment's going to be about. Okay, so imagine this. Imagine you have a history class and you are looking up information on, let's say, the Roman Empire. Okay, how would you go about that? Okay, so if you think on your history classes and the world history that you've had, or world cultures, how would you find information on that? Well, you have one way that you could use your, you could use your world history textbook. Okay, your textbook has accounts of information that have been handed down from generation to generation, and they've been put into a collection of a textbook. Okay. These can be everything from pictures of certain artifacts or places to actual copies of documents. Okay, all right. You can also go to places like the Museum of Natural History in New York, where there could be exhibits. You can actually see treaties, documents, signed copies of the actual works on display here. Okay, that's a great place to visit, and it can yield a lot of information. Okay. Lastly, you can look into talking to curators. Okay, this is Sandra Olson. She was the curator of anthropology at the Museum of Natural History in New York for some time. You could talk to her or one of her colleagues about a specific topic that you're looking to investigate on the Roman dynasty. Okay, so these are three ways that you can gather information for other classes, other courses, other topics, other subjects. But what this doesn't have, and there is a fundamental difference between this information's gathering and how we gather scientific information, is you really can't do experiments on this. You can't really run variables. You can't really run a process to look at any of this information and gather hard empirical data. Okay, so how does this vary from what we see in the sciences, biology, physics, ecology, chemistry, etc.? How does this differ? Okay, and what exactly was was the way that we came about it? All right, so it starts with this gentleman here. This is Aristotle. Aristotle came up with what's called the inductive-deductive method of scientific experimentation or methodology. Well, what this was is the inductive. You would make an induction or a conclusion based on an observation. Okay, you would see something and then you draw a conclusion about it. Okay, so from that you would keep looking, keep observing, and you would compare this initial observation to future observations, and then that would give you a much stronger base of knowledge to go on, and that would be the deductive side. That is where this becomes very what he called empirical. This was called empiricism because it relied on the five senses and the observations to gather your data. Okay, so from his information, and he was a very large contributor to most of what goes on in science in the early days, he greatly influenced this gentleman, Robert Gosseteste. Robert Gosseteste was heralded as the person, one of the first people to truly understand Aristotle's inductive, deductive, and empirical methodology. Okay, but what he furthered was that you verify this information and these observations and this reasoning through further experimentation. Okay, so this is where you start to reproduce and you start to cycle through and do it over and over again, and you change and adapt your hypotheses, your methods to get better, more reliable data. Okay, Even though he was a far cry off, he took a huge leap in the string and the lineage of this process. Well, Robert Gosse test went very largely to influence this gentleman, Roger Bacon. Roger Bacon was a friar in Europe, and he really looked at a repeating cycle of observation, hypothesis, experimentation, and then the need for independent verification. Okay? So by this independent verification element, what he was suggesting was that if there's a set process, then if Roger Bacon conducted an experiment, you conducted the experiment, I conducted the experiment, by using this same set of procedures, we could be in 3 different parts of the world experimenting on the same thing, but since our process is the same, we should get results that are consistent. So you can compare yours to mine and mine to Roger Bacon's, and we can get a much better idea of why those results came out the way they did. Okay, so that is going to be what he came up with and added to the science. Okay, so now that we know where it came from, we know that there's a difference between how scientific or the scientific method differs from how we gather information in other subjects like history. We can go on and now look at it in a little more detail. So what is the scientific method? Okay, scientific method, it's really a body of techniques. Okay, it's used to do the following: to investigate the phenomena. One that you have seen in an observation that you really don't know much about and it intrigues you. That's a phenomenon. Okay, it is going to be used to acquire new knowledge or correct and integrate previous knowledge into what you're looking at. Okay, so this is going to be a couple uses for this scientific method. Well, if you want to be more scientific about it, it's a method of inquiry based on empirical or measurable evidence. This measurable evidence is really your data that is gotten from the 5 senses. Okay, and this data is subject to the specific principles of reasoning that are laid out in your hypothesis or hypotheses, depending on how many you have. Okay, so when it comes down to it, guys, it's really about the observations, and from those observations, data, man, it's about the data. Okay, so let's take this and now let's look at it. Let's look at how these steps go in a linear fashion, and then I'll talk about this in more of a cyclic fashion. Okay, so here it is. So the first thing that's going to happen is going to be what you see up here. You ask a question. Okay, so when you look at this, you're going to ask a question. That's number 1. Okay, so by asking a question, you are going to already have had, at this point, you're going to have an observation. Okay, that observation is going to basically stick in your head, and it's going to develop a question. And it might be something that you don't sleep at night. It might be something that you pass every day, and every day it keeps becoming more and more important to you. So that's the first part. Next, what you do is you're going to ask that question. You're going to look at background information. You might do an internet search. You might go to the library. You might go to a specific college library, Google Scholar. You can use a bunch of online sources and do background research. Say, what's done on this already? What's been researched? Has this topic been researched so much that for you to get involved with it would be needless because the information's already out there? Okay, so that's something that it can be valuable for. The other thing is, what if nothing is put on this subject? What if you are going to be breaking ground on something that very few people have gone and studied? If that is the case, you could essentially be forging new ground. And that's where it becomes special. You might be on your own for a while, but in the end, what's going to end up happening is you're going to add to the scientific body of knowledge in a place where few, if no other people, ever have. That's a pretty unique thing. Okay, so from that area, when you have your background research, you have your question, you're really curious about this, your next phase is to go into what's basically called constructing a hypothesis. Okay, so now your hypothesis is really, this is in alternate terms, you probably have heard this, it's really just an educated guess. Okay, so what goes into this educated guess? All right, you've done some background research. you have an important question that's driving you into this process. But how is this supposed to look? So these are the statements that you guys have probably seen: the if, then, because. Okay? So really, let's say we're looking at something like plant growth. Okay? And if a plant receives less sunlight than it should, then it will have stunted growth because it doesn't produce the level of sugar that it needs to grow normally. Okay? So that is something that you would see as a hypothesis or an educated guess. All right? So once you have your hypothesis, this really dictates where your design for your experiment goes. Okay? So the next step would, in essence, be, when we look at it, number 4 here, is going to be test with an experiment. So before you test, you have to design an experiment. We are going to do this in our next round of this series, in our video 2. So I'll get a little bit more into that and variables. But once you develop an experiment and you go through it and you gather your data, so once you go through it, you look at it and say, is this procedure working? Well, if it is working, then that means if you have your hypothesis kind of in the back of your head on one side and you have the data or what you're looking to produce on the other, if that data doesn't necessarily speak to your hypothesis to say, okay, we support you or we have nothing to do with you, that means you can either follow it or you might have to tweak things a little bit to have one being more in line with the other. Okay? So if that's the case, then you just go back. You go back from that phase, you go back to the test with an experiment. Maybe you have to change your experimental variables. Maybe you have to change what you're observing or what data you're collecting so that it looks directly at your hypothesis to help support it or help reject it. Okay? So if it does, in fact, work and it does speak directly to the ability to support or reject it, then you conduct it. Go through it all the way to the end. Look at the data. Look at it with a fine-tooth comb. Undergo statistical analyses. Show it to friends. See what they think. Show it to professors. Show it to teachers. See what they think about it. Analyze it. And from that analysis, you're going to look for patterns. You're going to look for trends. And you're going to draw conclusions based on that. Okay? Now, once you've done all that, if your conclusions 100% align, so that analysis is step 6. So number 7, if your results completely align with your hypothesis, and your hypothesis is so supported by your data, then you are basically looking at, right now, the next step, which is going to be number 8, to communicate your results, which is really to write your formal paper or article on your experiment. Okay? And we're going to talk about the sequence and what to include in that article in the final installment, the 4th installment of this series. Okay? And then the next thing, what happens if those results don't at all support your hypothesis, actually helps reject it? Or if they partially support it and partially reject it, which leaves you confused, really not knowing what caused it or so forth. Well, so this is going to be another situation. Results only align partially. So if this is the case, all right, you can do a couple things here. One of which, you could make this information new background research to then go back and reconstruct a hypothesis again. And this could really serve at almost like what we call in science, we might actually even call this something like a pilot study, which really just goes towards making you more of an expert in that particular subject. Okay? So really, you're going back to the hypothesis at that point, and that's going to dictate you to change your experiment to develop a way to gather new and different or potentially better data that is going to give you then onto the pathway that leads you down here as opposed to down here. Okay? All right. Now, one thing I want you guys to know, and this is a very important point, your hypothesis does not always have to be supported. Okay? You have to remember, you've done this whole experiment, you've changed your variables, you've done all that. If you find out that everything is opposite of what you thought, that's still information. And the thing is, you could still find out why something happened. You can do a post-experiment research. Okay? You did a background research before, but you could do a post-experiment research on why this happened. Okay, maybe there's a study on what you found in your experiment that tested something else. Well, you could still communicate that in your results, and remember, you are still forwarding the knowledge on that topic. And someone in the future could then stand on your shoulders, being the giant, and make that subject really even more knowledgeable in the science community in the future. Okay, so that is what we're looking at here. So remember, if it's not supported or partially supported, you could find out why. And if you find out enough information as to why and you communicate that, that's almost just as good as being supported. It just means that your guess wasn't 100% right. And in essence, you're seeing information that comes out that maybe didn't surprise you, but that's unique, and that's cool. Okay? All right. So let's look at this on how it appears in a cyclical fashion. Okay? So if we look at this and we go up to our first stage, okay? So here we go. Number one, we're making observations. This is based on what you see, what's kind of tweaking your interest and really getting you going on the topic. Okay. From there, right? Thinking of the interesting questions. The patterns are arising, so you're like, okay, why is that happening? And saying, how can I find out more about this? Okay. So then, what you're going to do is you're going to start to look at and formulate hypotheses. Okay. What are the causes for this? This phenomenon that I'm wondering about. Okay, so once you start to formulate those, then you're going to really make your formal educated guess. Okay, that's going to be number 4 here. So these are going to be the actual final formal hypotheses or your testable guesses, testable predictions that you will then undergo your experiment to reject or to support. Okay, so after those testable predictions are made, here we go. We undergo our experiment. So this is the fun time. This is where we enjoy ourselves. We gather the data, conduct the experiments, we do our multiple trials, and we get data that is relevant. And by relevant, it's pretty much data that speaks directly to our hypothesis and helps us either reject or accept it. Now, if it doesn't, remember, then we go back here. Okay, so this is where we kind of have some trouble, and maybe we go back and we refine our procedures. Maybe we alter our methods. Maybe we reject our hypotheses, and maybe we change it and do a hypothesis. Maybe you do number 2, you do a second one. Maybe you produce a third one. Maybe you even produce a 10th one. Okay, this is all about cycling through and making sure the data align with the hypothesis. Sometimes it's necessary to do this, Okay, so if everything goes well and your data speaks directly to your hypothesis and is well supported, you can develop general theories. Now, there is a difference between a hypothesis and a theory. A theory is just a very, very supported hypothesis that have been done by multiple scientists in multiple places, maybe multiple generations, from different angles of the same topic, maybe even the same study on multiple different occasions in different labs, the same exact one down to the step, get the same exact results. That means it has more weight, it's more accurate, and it's even harder to disprove or to reject at this point. That's when we start developing theories. And in the ultimate case, that becomes a law when almost in 100% of the cases, it always comes out that way. That's when it becomes a law. Okay, so as you see, this is a cycle. So after you develop your general theory, you might see that same thing happen again. You might see it happen and you might observe it in another facet. Okay. Well, when that happens, well, there you go. So there you got your questions are starting to arise again. And when that happens, we start cycling through this again and again. Okay. So this is where and how we use the scientific method. So whether you're doing an experiment in the backwoods, whether you're doing experiments in a $1 million lab, you still have a set of directions that are going to make sure that your data is reliable and that you can compare yours to mine and mine to his, and we can actually have a conversation about why the differences happen or why they were exactly the same. Okay. Well, I hope that was helpful, everybody. Tune in next time for episode 2, which is experimental design, identifying variables. Take care.